First of all, I'd like to thank OGT for giving me the opportunity to come and speak with you today. And I'd also like to thank my colleague, Dr. Capacicu, for a wonderful presentation this morning. Today, I'm going to be talking about some of our experiences with the myeloid panel NGS sequencing in our laboratory. And I'd like to start by thanking some of my colleagues who've been very critical in that process. Darcy Butcher, Landry Nfonsom, Barry Ng, and Sean Pajak, who are all involved in this project, as well as our fabulous technologists in the lab who are working on a number of different projects. So the first time, I remember the very first time that I looked down a microscope to see chromosomes from a patient with AML. And I'd seen chromosomes before at the microscope, but never so complex and, well, shall I say, unrecognizable as they were in that single patient. And of course, we now recognize that the significance of that complexity is it's a poor prognostic factor in AML. But there's so many different recurrent cytogenetic abnormalities that we now recognize as being clinically significant. However, what's really exciting, I find, albeit the chromosomes are the things that truly excite me, there's these smaller genetic abnormalities that we can't see down the microscope, and increasingly we're becoming aware of their diagnostic, prognostic significance, and even more and more importantly, their roles as targets for therapeutics. And so that genetic information at the gene level is also becoming equally as important as the cytogenetics that we've always looked at in MDS and AML. So to investigate some of these genetic mutations, our laboratory launched a Myeloid NGS panel in 2017. We looked at 27 different genes in our bioinformatic pipeline, and these are the locations of those genes. Most of these genes, well, actually, all of these genes were recommended through Cancer Care Ontario for analysis in patients with complex malignant hematology, including MDS and AML. But after we had launched that assay, we also did a continuous quality improvement project where we decided to compare our results from the NGS panel that we were currently using to an OGT SureSeq Myeloid panel. Sorry, didn't mean to mess up that name. And ultimately, we were looking at the exact same genes, but there were some very different important differences in the chemistry that we wanted to see. Does that affect our ability to detect some of our problematic regions, particularly in CEBPA, which was high GC content, and we were struggling trying to get good coverage of that gene. And there were some other gene regions that we were having particular concerns about. The two panels covered all the same genes, but the panel we were originally using had an amplicon-based chemistry, whereas that from OGT was a hybridization capture-based chemistry. We were recommended we could use 12 to 16 samples on the SureSeq, but we do find we are currently running 8 samples with each batch. As part of that validation, we did look at some reference controls from that had a number of different genes with expected variant allele frequencies ranging between 5 to 10%. And we were able to detect all of these variants using both panels with variant allele frequencies that were in appropriate ranges. We also assessed, there was 48 specimens that we were included in this validation study. We looked at 1,187 variants across the total genes that we were examining. In dark green represent pathogenic genes or variants, and the light green are variants that are benign and not reported on our reports. In doing so, we had 100% sensitivity for all of the variants that were detected by the two platforms and a specificity of greater than 99%. There was several variants in CEBPA that we did pick up by Sanger sequencing, and weren't able to see them on the other NGS panel, but we were able to see them on OGT SureSeq, which was very reassuring. And this is the comparison of this being the gene that we were particularly concerned about, comparing it between the two NGS panels. On the top is the raw data from OGT, and on the bottom is from the other gene panel that we were using. And you could see on the bottom that there's several regions where there's gaps of coverage, versus with the OGT, we had very, very even coverage across the entire gene. This allowed us much easier to pick up variants regardless of where they were. We also saw, for a number of important pathogenic variants, we were able to detect them using both platforms, but with the OGT, we did have much more consistent coverage across the regions that we were interested in. This is an example here of a TET2 variant with a 2 base pair deletion. We have since gone on. We are now using this with the larger cohort of patients on a much more regular basis. And we've so far sequenced, the time of putting this together a few weeks ago, 265 AML patients. And this is the profile of the mutations across those 27 genes. It's very typical of the profile that we would expect to see based on the literature, with NPM1 and DNMT3 mutations being most common, as well as various other genes. This is also a problematic region for the NGS, and we do see that the FLT3 ITDs are being pulled out very easily using this platform. This is an example of 3 different ITDs that we have seen in our cohort, with ranging variant allele frequency as low as 4% and as high as almost 30% on these 3 cases, but also including a very large internal tandem duplication of FLT3 that's 162 base pair duplication that the platform was very easily able to pick up. And these have all been confirmed by single analyte testing as well as by PCR and electrophoresis as well. We also wanted to assess, is the variance that we are detecting, does it coincide with what we expect from the literature? So in that same AML cohort, we did go into the literature to try and assess, well, what is the expected frequencies? So I've shown in purple the range of frequencies that we were seeing expected in the literature. And this has somehow gotten shifted. I don't know what happened here. I apologize, but they were overlapped. In general, what you were supposed to be seeing is that the frequencies of what we were seeing in these patients coincided in general with what we expected for the literature. Our FLT3 and NPM1 mutations were a little bit low, but again, we also do those by single analyte testing, and compared to the previous platform that we were using, that was also low in there. So we just think there's something happening perhaps in our population where those are a little bit lower than we would expect, but it is still in the normal range. We were also concerned about ASXL1 because there's some literature out there that the most common variant can occur as an artifact. So we do restrict when we're reporting ASXL1 variants, particular of that common duplication, we only report it when the variant allele frequency is above a certain threshold. We're seeing in this cohort that our frequencies were a little bit higher than what we would expect from the literature. So we wanted to take a closer look at that. This is a summary of all the ASXL1 variants that we saw using both platforms. And this is a very, very similar mutation profile regardless of the platform that we used. And if you consider the number of mutations at each locus between using either the SureSeq or the NGS panel, those patterns again were similar. So we don't think that this is a technical artifact, that our numbers are a little bit higher, and again, just may reflect the local cohort that we are testing. We did a similar analysis in MDS, where we looked at the profiles of variants found in our patients. Our cohort in this at this time is 113. We are prioritizing our AML analysis, so we have more of our cohort being the AML. We have sequenced just about the same number of MDSs, but haven't quite got through all those analyses. But again, these profiles are very similar to what we'd expect in the literature. And this slide has also got shifted, so I apologize. But ultimately, again, when we compared to the literature, we were finding very similar frequencies to what we'd expect in the literature. So that was reassuring that the quality of the variants that we're detecting is good. We did notice with the other panel that some of our frequencies were high compared to the literature, including RUNX1 and TP53. And I think that was partially due to a little bit more noise that we were picking up before, and that issue seems to be resolved with OGT. So we're confident now that we are reporting appropriate variants. But I think what is more interesting, at least from a lab geneticist standpoint, is the individual patients. We can make conclusions about our cohort. We say, yes, they're in line with what's happening in the literature, but really we are not examining cohorts, we are examining individual patients, and it's individual patient stories, I think, that are what excite me more. I feel for these patients, but I want to understand better what's happening in those individuals, because ultimately that's how we can better provide care for these patients. And this is something coming from the cytogenetics world. When you're looking at individual cells, you can see these patterns of clonal evolution. We have a cell that's got +7, we've got 8 of those, and now we've got 5 cells that have an extra copy of 7 and an extra copy of 11. That's clearly clonal evolution. It's harder when you're looking at DNA that's all kind of jumbled into a tube, but we do have instances where we can kind of see that clonal evolution happening too. This is a case where we have CF3B1 and EZH2 mutations, and we have a higher frequency of the CF3B1. So you can imagine that was the first mutation that happened, and then we've acquired EZH2 mutations as a part of clonal evolution. But this is more kind of making me think of that first patient and when I saw underneath the microscope, where we're just seeing these crazy amounts of mutations. And this is when you start to worry about these patients and what's going on, that they have so many mutations in a single individual. But we can glean things from them. We've detected ASXL1 mutations and RUNX1. We know those are generally associated with a poor prognosis, and so this patient may have a worse course of disease. But we're also seeing things like FLT3 mutations, and we now know that there are therapies available for FLT3, so that this type of information can be useful to try and treat this patient most appropriately. This is some of the examples. I tried to pull out examples of cases with it. I kind of had to scratch my head about. This was an individual who came through our laboratory, 77-year-old male with an abnormal karyotype of trisomy 8, which is generally considered a good prognostic factor. Flow cytometry noted changes that are sometimes observed in MDS. Later on, a few months later, we did do testing again, and the trisomy 8 was persistent, and there's no additional chromosome abnormalities. But there was this very, very weak positive FLT3 variant when we were doing single analyte testing, and that's shown right here. So this is a restriction enzyme assay that we do to target the tyrosine kinase domain to see if they have variants of that particular domain. And we do know if there are variants present, then there are therapies available that can be used to target that. When we did the NGS sequencing, however, it was a very atypical FLT3 mutation of the tyrosine kinase domain. It was an insertion deletion event involving the amino acids right next to that particular codon of relevance in the tyrosine kinase domain. And we think that this is, in fact, what's impacted the ability to detect the variant by the restriction enzyme assay. It clearly is being picked up by the restriction enzyme assay, but by NGS, it's only 2.8%. So the fact that we have a very, very weak peak is consistent with what we're seeing with NGS. The same patient, however, also had atypical FLT3 mutations in the same region that we would normally see internal tandem duplications. So there was an insertion deletion event with a variant allele frequency of about 10% that results in a single amino acid gain. And that single amino acid insertion in the juxtamembrane protein domain is predicted to result in gain of function of the FLT3 protein, albeit it's considerably smaller than the ITDs we normally see. In addition to that, we also identified a missense change also in the juxtamembrane domain. We've never seen this particular mutation in our lab, but there is a report out there that this mutation does result in a gain of function change. They are all on different reads, which indicates that these are not in the same on the same allele. They're either in different alleles, in the same cells, or in different cell populations, and we're unable to discern that. But it's interesting because this patient now has 3 different FLT3 mutations. So clearly, we can't have 2 mutations in the same patient if they're all on the same, all on different alleles. We only have 2 alleles available. So there's something else going on here. We've got multiple clones. Is it the same disease? Is it progression? Is it different subclones of an initial assault? Or what's happening in these patients? This is not the only case where we've had multiple mutations in the same gene and same patient. This is an example of an individual with multiple WT1 variants. We have 3 different variants in this individual here, ranging in allele frequency from 5.7% all the way up to almost 40%. And they all occur on different sequence reads in our NGS, again indicating that these are either in trans, in cis, we can't tell, but because there's 3 different populations, obviously there's more than one subclone that has these mutations. Another example, we have a few of these in our lab too, of where there's multiple NRAS mutations. This is an 88-year-old female with otherwise normal karyotype and no apparent mutations in any of the single analyte testing that we did, but several variants by NGS, including 3 different NRAS mutations. And again, all on different sequence reads, so these are happening in different alleles. Just an example of some of the other ones. This is only 3 of the cases. We do have other examples where we have multiple NRAS variants. And it begs the question of what's happening in these cases. Is this hematopoiesis, particularly when a lot of those variants are at very, very low levels. But how do we know? Particularly with the NRAS, these tend to be later events, but could there be solid tumors in these patients? And we're picking that up because we are looking at blood. Sometimes you can have cell-free tumor DNA. Not really clear, and unfortunately, in these individual cases, it's going to be very hard to pull that information out. But we are looking at trying to figure that out. So in conclusion, I think I hope that I've shown that there's comparable assay target enrichment for the regions of interest, but we do have, clearly we've got improved uniformity of coverage using the SureSeq libraries for this myeloid panel. We've been able to detect variants of allele frequency at 5% or greater, and the prevalence of variants detected in AML and MDS are similar to those that we'd expect for the literature. But that NGS sequencing enables us to detect variants that may refine the risk stratifications or provide information about therapies that may be useful in these individuals. But ultimately, there's these unique cases that we still don't understand. And I think these cases highlight for us there's a lot of biology that we still have to learn regarding AML, and hopefully in the future this will lead to improved diagnostics and therapeutics for these patients. Thank you.